Hello biologists, and welcome to your next screencast. Today we're going to talk about Chapter 7, DNA Structure and Replication. In this chapter's case study, we'll look at how we can use DNA evidence and DNA fingerprinting to prove innocence or guilt in the court of law. The chapter case study focuses on the Innocence Project. The Innocence Project was founded in 1992 at Yeshiva University's Cardozo School of Law in New York City. The Innocence Project uses DNA evidence to free people wrongly convicted of crimes. DNA evidence was first used in a court in 1985. Today, DNA testing is a standard part of court cases. DNA evidence is an extremely accurate way of matching crime scene evidence to perpetrators. The photo shows Roy Brown. In 1991, he was convicted of homicide. He spent 15 years in jail until the Innocence Project proved his innocence using DNA evidence. The Netflix series Making a Murderer is about Stephen Avery, a man who was wrongly convicted of sexual assault and attempted murder. He spent 18 years in jail until the Innocence Project helped free him. DNA stands for deoxyribonucleic acid. It's normally just referred to as DNA. DNA is a genetic or hereditary molecule passed from parent to offspring. It's found in all living things, from the smallest bacterium to any plant to any animal. It's found in all living things because it serves as an instruction manual for how to build an individual organism. The DNA found in every cell of an organism is known as the organism's genome. DNA is found in the nucleus in eukaryotes and freely floating in the cytoplasm in prokaryotes. DNA exists in the form of chromosomes, single DNA molecules wrapped around proteins. The diagram shows how chromosomes are structured and found in the nucleus of eukaryotic cells. So DNA can be organized into chromosomes. We can break a chromosome down into genes. Each gene on a chromosome has instructions to make a protein. Try and use the diagram on this slide to distinguish between a chromosome, a gene, and DNA. Think of genes as recipes, and each gene recipe makes a protein. Humans share a surprising number of genes with other organisms. 18% of our genes are the same as a plant, and 98% of our genes are the same as a chimpanzee. This infographic looks at chromosomes in human cells. Each human cell has 46 chromosomes, or 23 pairs. The 23rd pair determines our sex and are labeled the X and Y chromosomes. As I just mentioned, humans have 46 chromosomes in each cell, or 23 pairs. One chromosome in each pair comes from your biological mother. The other chromosome in each pair comes from your biological father. This image is a karyotype. It's made by taking photographs of your chromosomes under a microscope and organizing them by pair. The 23rd pair of chromosomes are known as the sex chromosomes. They determine a person's biological sex. A person with two larger X chromosomes is biologically female. A person with one larger X chromosome and one smaller Y chromosome is biologically male. You can see the sex chromosomes of a person who is biologically male in the lower right of the karyotype. Unless you have an identical twin, your DNA is unique. We need to know something about the structure of DNA to understand why. Remember that DNA is a polymer made up of repeating units called nucleotides. Each nucleotide is made up of a sugar, a phosphate, and a base. If you think of DNA as a twisted ladder, then the sides of the ladder are made up of sugar and phosphate, and the rungs of the ladder are made up of bases. In every DNA molecule, two strands of nucleotides pair up and then they twist around each other to form a spiral-shaped double helix. Sugars and phosphates form the outside of the double helix, or the backbone. Bases form the internal rungs of the double helix. The two nucleotide strands of DNA are held together by hydrogen bonding. The hydrogen bonds form between specific bases. This is called complementary base pairing. A will always pair with T, and C will always pair with G. In the diagram, you can see base pairing occurring, with A pairing with T and C pairing with G, holding the two sides of the double helix together. Every person has a specific and unique sequence of nucleotides in their DNA. To give you an idea of how big our genome is, humans have about 3 billion base pairs found in our 23 chromosomes. To study our DNA, we can extract DNA from cells and create a DNA profile. A DNA profile is a visual representation of a person's unique DNA. In order to make a DNA profile, we need to make many copies of the DNA of interest. Before we talk about how DNA is copied in a lab, let's talk about how it naturally happens. When DNA makes a copy of itself, it's called DNA replication. DNA replication takes advantage of the complementary base pairing rules that we talked about earlier, A always pairing with T and C always pairing with G. First, the hydrogen bonds that hold the base pairs together have to be broken and the helix has to be unwound. The animation on this slide shows how this is done by an enzyme called helicase. After the DNA is unzipped and unwound, another enzyme comes in called DNA polymerase. DNA polymerase reads each strand of DNA and adds the complementary nucleotide using the rules of base pairing. For example, if it sees an adenine nucleotide, then it'll add a thymine nucleotide. When DNA replication is complete, you end up with two identical strands of DNA. DNA replication is known as semi-conservative. That means that in each strand of resulting DNA, you have half old DNA and half new DNA. You can see that in the diagram on this slide. In the resulting strands of DNA, half is old blue DNA and half is green new DNA. In a lab, we can copy DNA by using a technique called the polymerase chain reaction, or PCR. In PCR, a specific DNA segment is replicated and amplified. To replicate means to copy. To amplify means to make many copies of. In order to do PCR, we need primers. Primers are short segments of DNA that guide DNA polymerase to the section of DNA we want to copy. The diagram shows what we need in order to do PCR: the DNA of interest, nucleotides, DNA polymerase, and primers. PCR has 3 steps: heating, strand separation, and DNA replication. Each time these 3 steps happen is called a cycle, and after every cycle, you double the amount of DNA you have. Heating separates the strands of DNA and allows DNA replication to happen. Through PCR, we can replicate DNA many times through cycling. We can make billions of copies from just a few molecules of DNA. The diagram shows how that after every round or cycle of PCR, we effectively double the amount of DNA. After we have enough DNA through PCR, we can create a DNA profile. DNA profiles are important because aside from identical twins, no two people have the exact same DNA sequence.